Hello friends, so in this lecture we will be dealing with the DNA replication. Okay. DNA replication is the process by which DNA makes its own copy. Makes its copy. Suppose that this is the DNA we have. When it replicates, it becomes double. Okay. A copy of this DNA is created. This is known as replication. Okay. Replication means replica is nothing but a copy. Replication is generation the copy. Simple. Okay. So as we all know that Watson and Crick, what did they do? They gave, or you can say that proposed, gave double helical model of DNA. What is the double helical model of DNA? It means that okay, it means that DNA. Okay, there are two strands of the DNA, and they are coiled like a helix. Okay, two strands of a DNA run parallel to each other. They are joined with each other by hydrogen bonds, and they are coiled like a helix. Okay, and we also know that inside the DNA we have a polarity also. This is a polarity of the DNA. Okay, suppose that if we are considering this one strand, I'm just making it thicker so that it is easy to identify. Okay, it is easy to differentiate between the two strands. Okay, suppose that here it is the 5 prime end of this one, and so here it should be the 3 prime end. Okay, this is polarity of the DNA. If you have studied the structure of the DNA, then you might already be aware of this. And inside the opposite strand we have the opposite polarity. So in front of 5 prime here we have 3 prime, in front of 3 prime here with the 5 prime end of the opposite strand. Okay, they are joined together by hydrogen bond. This we already know. And one more thing is that there is a phenomena of what? Of base pairing. Of base pairing. Okay, what is that phenomena of base pairing? That means that okay, so basically we have okay, so basically nucleotides we have two types. Okay, purines, pyrimidines. Okay, purines we have adenine, guanine. Pyrimidines we have okay, cytosine, uracil, and thymine. This uracil is only in RNA, thymine only in DNA, and this A, G, and C are present in both RNA and DNA. Clear? Let me just take one more page. Okay. So according to the phenomena of base pairing, adenine base pairs with uracil if it is RNA, and thymine if it is DNA. If we are synthesizing DNA, adenine can base pair with thymine. If we are synthesizing RNA, then adenine can base pair with uracil. Similarly, guanine will base pair with cytosine. And the opposite also you can say that guanine base pairs with cytosine, or cytosine base pairs with guanine. Same is the case for A, U, and T. Okay. So this is the phenomena of base pairing. On the or using all these concepts. Okay, Watson and Crick, they said that okay, or you can say that they proposed the scheme for a replication. Okay, mainly on the phenomena of base pairing. Okay, so how did they propose? Okay, so what they did is that they told that suppose that here we have the DNA. I just drawn it in a simplistic way. Okay, these two strands we have, and they are joined together by hydrogen bonds. Okay, or let us just consider it. Okay, let us be more realistic. Okay, suppose that here we have adenine, here we have G, here we have T, and here we have C. Okay, so in the opposite stand we are going to, what are we going to have? Okay, let me just make you clear that A can base pair only with what? I've told you just now. Okay, A can base pair with only with what? Okay, or G can base pair with only with what? Okay, so G can base pair with C, A can base pair with U or T. Clear? A can base pair with U or T, G can base pair with C. U if it is RNA, T if it is DNA. So in front of A, what are we going to get? We are going to get T. In front of G, we are going to get C. In front of T, we are going to get A. In front of C, we are going to get G. And they are joined together by hydrogen bonds. Simple. So suppose that we cut this DNA in two parts. We have just cut this DNA in two parts. Okay, and separated them. Okay, and separated them. Yeah, just a second. Yeah, so I've cut this in two parts and separated them. Here we have T. Okay, T. So you have cut them in two parts and separated them, and I am just telling you to synthesize a new strand in front of them. Okay, what are the base pairs that you are going to put? What are the base pairs that you are going to put here? In front of A, what can you put? Can we put G or C, C or G, or even, or U? We cannot put U because it is DNA. We cannot put G or C because A cannot base pair with C. A can base pair with only with U and T. U if it is RNA, if we are synthesizing RNA, T if we are synthesizing DNA. So we will have to put T here. Similarly, here we will have C, here we will have A, here we will have G. Okay. Similar on the other stand, here we are going to have A, G, T, C. Clear? Surprisingly, let me just number them. Suppose that this is first, second, third. If you are seeing first, second, and third and comparing them, you can very easily say that first, second, and third are identical. What have we just done now? We have just replicated the DNA. What is replication? Replication is just synthesizing the exact copy. We have just synthesized the exact copy of the DNA using the phenomena of base pairing. Okay. And one more thing that I want to say here is that this method of replication is called as semi-conservative mode of replication. What do we mean by semi-conservative mode of replication? Semi-conservative, okay. Semi means half, conservative. It means that if you take the daughter DNA and see its one strand, you can very easily see that it is derived from parent. It is paternally derived, or you can say that derived from parent. Simple. Derived from parent. While the other strand is newly synthesized. Same is the case here. Semi-conservative means half conserved. Conserved means basically it wants to say that it is conserved, means it is derived from the parent. Half conservative means half it is derived from the parent and half is newly synthesized. This is known as semi-conservative mode of replication. And let me just show you some other ways by which it could have been done. Okay, which it doesn't happen actually. So suppose that here we had the DNA. Okay, we just split it into two. Okay, synthesized new strand. Okay, and what we did is that we just separated them again. We separated them again. Okay, okay. Suppose if we just separated them again, okay, and then rejoined them like this. In front of white, I've joined white. I've joined white with white, and green with green. Sorry, green with green. Can you say that this is semi-conservative? Not at all, because one of them is completely derived from the parent, while the other is completely newly synthesized. This green I have represented by the newly synthesized strand I have represented by the green. So one is completely derived from parent, and one is completely derived from one is completely newly synthesized. This does not happen at all. Actually, this is what happens. Semi-conservative mode of replication is what happens. How can we say that this happened? For this, we did several experiments. Let me just explain few of them. Okay, so experimental proof for, okay, experimental proof for semi-conservative mode of replication. Okay, experimental proof for semi-conservative mode of replication. How did we experimentally verify this concept? Okay, how did we actually verify this concept? Okay, it was done by, okay, first experiment was done by Michelson and Stahl. Michelson and Stahl. What did they do? Okay, just a second. Okay, so what did Michelson and Stahl do? They took E. coli. Okay, and let me tell you one more thing, my dear students, that in DNA we have nitrogenous bases. If you are, even minimally aware of the structure of the DNA, then you should know that DNA does have nitrogenous bases. Okay, and by the name of the nitrogenous bases, you can clearly say that these nitrogenous bases contain nitrogen. Contain nitrogen. Thus, we can say that DNA contains nitrogen. Simple. Clear. So he took E. coli. They took E. coli. Okay. They grew it in a medium. Okay. Suppose that this is the medium. Okay. In which there was only 15 NH4Cl. 15N is the heavier isotope of nitrogen. Isotope of nitrogen. 15N is the heavier isotope of nitrogen. So they took E. coli into this medium. Okay, into this medium. And after that, what did they do? They just grew it. Okay, they grew it in the medium containing 15 NH4Cl. Okay, so this bacteria is growing and growing, and it is utilizing 15N as its source of nitrogen. Okay, so if you take sample after some time, from the culture that you have just grown, you are going to observe that the E. coli contain 15N in their DNA. Earlier they had 14N. This is the normal isotope of nitrogen. But if you are growing it on a medium containing 15N NH4Cl, then you are going to observe 15N in the DNA of the E. coli. Clear? Okay. Okay. This is the prerequisite knowledge for you. Okay. After that, they grew it back. They took the they grew the sample, they took the sample and grew it back into the medium containing 14 NH4Cl. Okay, 14 NH4Cl. Clear? 14 NH4Cl. What happened then? Okay, and then okay, they took samples after every 20 minutes. They took sample, then after 20 minutes they took sample. What did they observe? I will just tell you in just a moment. Okay, let me just tell you one more thing. Okay, this just keep in your mind the overall process that we are doing. I just want to tell you one concept, that you can separate this 14N and 15N containing E. coli by what? Since there is a difference in their weight. Okay, 15N containing E. coli is going to be heavier than the 14N containing E. coli. So we can separate it by centrifuge, and that also CSCl density gradient centrifuge. Clear? So if you took the samples in this test tube and just run it through it, the bacterium containing 15N will be at the bottom, while those containing 14N will be at the top. Okay, somewhat above the bottom. Clear? So this is how we can differentiate the bacterium containing 15N, 14N. And one more thing is that, suppose that there is a bacterium, this bacterium contains both strands. DNA is double stranded, my dear students. DNA is double stranded. Suppose that this strand is 14N, this is also 14N. There is also a possibility. Okay, so the other bacterium we are having is the one containing this also 15N, this also 15N. There is one more possibility, my dear friends, that there is a bacterium, this contains one strand as 14N and the other strand as heavier one, 15N. Okay, these are the three possibilities that we can have. Can you just tell me where will this bacterium be found inside the density gradient centrifuge? Where is this bacterium going to be found? This bacterium is going to be found in between these two bacterium, because its density its density is going to be its weight, you can say that, is going to be the average of these two bacterium, the one containing 14 and the one containing 15N. This bacterium is going to have the weight as the average of these two. So inside the density gradient centrifuge, you are going to find it in between these two bacterium, in between the bacterium containing 14N and the bacterium containing 15N. So this is how we can separate them. Okay. So what happened is that, so let us first learn what they did. Suppose that we are just observing. Okay, we're just observing. We are here to verify. Why are we doing all these things? We are doing all these things so as to verify what? So as to verify the semi-conservative mode of replication. And what does semi-conservative mode of replication says? It says that in the daughter DNA, half of the DNA is derived from the parent and half of the DNA is newly synthesized. We are going to verify this. So just we are just going to observe. What are we going to observe? We are just going to see. Okay, so we were here, we took 14 N containing E. coli, grew it in the medium containing 15 NH4Cl. Okay, 15N gets incorporated inside the E. coli. We grew it back on the normal medium and we are taking samples after every 20 minutes. Okay, so we had the bacterium of this one. this one. Okay, and let me tell you one more thing is that the replication time, or not replication time, the duration of cell cycle, or you can say cell division. Cell cycle is better process. Cell cycle in E. coli is 20 minutes. Means in 20 minutes, one E. coli becomes 2. Okay. Suppose that you took the sample at t equal to 0. We all have this, all the E. coli containing this green DNA, this 15N DNA. You just ran it on centrifuge and you are observing things like this. You waited for 20 more minutes, took the sample again, and ran on the density gradient centrifuge. Okay, I just drawing the test tubes. Okay, so what we observed, what Michelson and Stoll observed during this experiment is that they observed bacterium somewhere like this. The bacteria were observed here in this location. First, the bacteria were observed in this location, then the bacteria were observed in this location. Okay, then they waited for 20 more minutes. Okay, they ran the centrifuge. They observed bacteria here and here. Okay, at 2 points, here and here. This is what they observed. Now let us try to link this. This is what they observed. Okay, this is what the experiment says. If you took the bacteria at t equal to 0, you are going to observe the bacteria here. After 20 minutes, you are going to bacteria observe here. After 20 minutes, you are going to bacteria observe here, here, and yeah, this color is better. After 20 minutes, you are going to observe bacteria here and here. Clear? Okay. This is what we are observing from the experiment. Now, this is our rule. This is our, semi-conservative mode of replication, theoretical thing, that half of the DNA is paternally derived and half of the DNA is newly synthesized. Let us try to link these two things. Okay. So I'm just drawing what the semi-conservative mode of replication would say, and we will just compare these two things. According to the semi-conservative mode of replication, suppose that this is the DNA. Okay, it replicates. Okay, 20 minutes. This paternal will split into 2, and new strands will be synthesized on this. Because now, see here the DNA is green one. Now why white ones are being synthesized? Because we have changed the source. Our source currently is 14NH4Cl. So 14NH4 gets incorporated inside the bacteria each time it is dividing. Okay, so we observed this. After 20 more minutes, my dear students. Okay, so this paternal will split. Paternal DNA will split like this. Okay, and new strand will be synthesized around it. White one. Okay, clear. So just compare these two. Green one, it is going to be the heaviest one, so you are going to observe it at the bottom. This one, you are going to observe it in the midway, somewhere in midway. This one, this and this and this, you are going to observe in the midway, somewhere in the midway, and this you are going to observe at the top. We have just verified the semi-conservative mode of replication. Okay, this experiment was done by Michelson and Stahl. Clear? Yeah. One more thing that I want to mention here is that Taylor and colleagues they did experiments on Vicia faba, which is also known as faba beans. Okay, and they said that DNA in chromosome also replicates semi-conservatively. Okay, what does this mean? Suppose that this is the DNA of a cell, and this cell is just prepared to divide. Okay, so the cell is just going to divide. No, yeah, the cell is going to divide. So for division, before division, the cell will replicate its chromosome. The cell will replicate its chromosome, or you can say that DNA as well. You can say, okay. So for that, when that will happen, the chromosome that we are going to observe will be somewhat like this. Okay, and you will observe something like this. The newly synthesized, if we are representing with green one, then you will observe things like this. Green one is newly synthesized. This white one is paternal or derived from parent. Clear. This experiment was done by Taylor and colleagues. Clear. That was it. And now talking about the mechanism of the DNA and various enzymes involved in DNA. Okay. So machinery and enzymes involved in DNA replication. Okay. What are the machinery and the enzymes involved in replication? So talking about prokaryotes, okay, main enzyme that we have is DNA dependent DNA polymerase. Why is it called so? Why is the name of enzyme so long? Okay, DNA dependent means the enzyme uses DNA as template. DNA polymerase is the enzyme synthesizes DNA, or polymerizes DNA. You can say that the enzyme synthesizes DNA. So overall, the enzyme uses DNA as template to synthesize DNA. This is what we were doing earlier. This was the DNA. We split it into two. Okay. And then the enzyme comes. It uses this as a template to synthesize new strand. This as template to synthesize new strand. This is what this enzyme do, and this is why such is the name of this enzyme. Okay. So the speed of okay, the speed of this of the enzyme is very fast. Okay, it is around 2000 base pairs synthesized per second. In 1 second, this enzyme is synthesizing 2000 base pairs. So it is very, very fast. Clear. Now talking about how what are the things that act as substrate for the synthesis of the DNA. Suppose that this is the new strand that we are synthesizing here, that you can see here. This is the new strand that we are synthesizing here. What are the substrates for this? What is the substrate acting for this? So the substrate as well as the energy source are the same, surprisingly same. Okay. So substrate and energy source, they are same, and they are deoxyribonucleic acid triphosphates. Okay, deoxyribonucleic acid triphosphates. So the normal structure of a nucleotide is this. Okay, let me just tell you one thing, that DNA is a heteropolymer. Heteropolymer means the constituting units are not the same. Means many different types of units join together to form a polymer. So DNA is a heteropolymer of what? Of nucleotides. Or you can say that, to be more specific, deoxyribonucleotides. Okay, and the structure of the DNA of the nucleotide is somewhat like this. This is a pentose sugar. Here we have the nitrogenous base, and here we have phosphate group. Okay. Since it is a triphosphate, we are going to have two more phosphates. Okay, so this is the overall structure of deoxyribonucleic acid triphosphates. Okay, in this structure is of nucleotide, which will act as the substrate. You can say that it is the basically the it will basically polymerize to form DNA. And the other remaining structure, here you can see two bonds, phosphate phosphate bonds, and they are energetic bonds. And these bonds will release energy, and this energy will be the source for the synthesis of the DNA, for polymerization of the DNA, you can say. Clear? So the source of energy as well as the substrate is the same, deoxyribonucleic acid triphosphates. Clear? Okay, so suppose that this is a cell. Okay, this is the DNA of the cell, and here replication is happening. Replication is just going on, and we have just zoomed in into this, and we are just observing it. Okay, so if you observe a replication, if you observe a replication, you will see somewhat like this. This is the point where replication is happening. Okay, I've just zoomed this point. Let me just represent it in a better way. Okay, so we have just zoomed this point. This is better, particular point here, and we are observing here. Okay, so the strand of the DNA, we already know that it is here it should be 5 prime, 3 prime. It has a polarity, and the opposite strand is of opposite polarity, 3 prime, 5 prime. Clear? Suppose here we have the our enzyme, DNA dependent DNA polymerase, DDTP in short, DNA dependent DNA polymerase. Okay, so this enzyme is running like this, and polymerization is happening in that direction. Okay, fine. Here, so the newly synthesized DNA have also to be moved in this direction. Clear? In this strand, okay, one more thing that I forgot to mention you here, that I just want to tell you that this DDRP, DDDP, DNA dependent DNA polymerase, it polymerizes only in 5 prime to 3 prime direction. Suppose that here we have a DNA. Okay, suppose that it is synthesizing DNA like this, in this direction, from this direction to this direction, it is synthesizing. So this must be the 5 prime end, and this must be the 3 prime end. So if we are observing here, if you are observing here, okay, here we have the 5 prime end. Opposite to the 3 prime end, it should be the 5 prime, simple. the two strands are always opposite inside. So here it is polymerizing, and here should be the 3 prime end. Okay, so here it is polymerizing from 5 prime to 3 prime. We have no problem with that. It can easily polymerize from 5 prime to 3 prime. It has to polymerize from 5 prime to 3 prime. It cannot polymerize from 3 prime to 5 prime. So let us just observe the opposite strand. What happens here? Okay, okay, okay. Let me just, yeah. Let me just show you by this highlighter. Okay. So here we have to synthesize like this. Here we will have the 3 prime end. Okay, here we will have the 3 prime end, and here we will have the 5 prime end. Can this DNA dependent DNA polymerize like this? The answer is not at all. It cannot polymerize from 3 prime to 5 prime. It can only polymerize from 5 prime to 3 prime like this. But replication is happening in this direction. Replication is happening in this direction. This enzyme is going in this direction. Can you understand the complexity here? The enzyme is going in this direction, but we have to polymerize in this direction. So what does this enzyme do? This enzyme first polymerizes from this direction, some fragment like this. It comes back, polymerizes like this, comes back, and polymerizes like this. This is how the enzyme compensates for this. Okay, so in this strand, the polymerization will be somewhat like this. Here we have the 5 prime. Just mention it here, 5 prime. It will be polymerizing in fragments, small fragments, and these fragments we call them as Okazaki fragments. Okay, and these fragments will later be joined by DNA ligases. Clear? These fragments will be later be joined by DNA ligases. Okay, so one more thing that I want to tell you that this strand, we call it as a leading strand, and this strand we call it as lagging strand. Okay, leading strand is we can also say it as continuous strand, because it is being synthesized continuously. Continuous strand. And lagging strand, we can say it as discontinuous strand. Okay, so that was all about DNA replication. Okay, I hope you enjoyed this lecture. Thank you so much. If you liked the video, then please do share with your friends. And if you want me to explain any topic to you that is quite difficult for you to understand, then please do let me know in the comment section, and please do share with your friends. I've just started this channel, and I don't know how this goes. If it goes well, then I do hope that it will go well. If it goes well, then I will just continue to upload as many lectures as you want, and it is really my passion to teach. This I have discovered just now, and then I just started to record the lectures. Okay, so thank you so much. If you like the video, then please do support, and yeah, see you.